Okay, Chapter 22, Anaerobes. Again, check over these objectives. We talked about anaerobes pretty well during our activity, so review the basic characteristics of anaerobes. Anaerobes are found everywhere, all over our body. They can cause diseases, all kinds of different diseases, brain abscesses, genitourinary tract infections, myonecrosis, sinus and dental infections, aspiration pneumonia. So any kind of infection really can be caused by an anaerobe, and they're found everywhere, all over the body. Some predisposing conditions, any kind of activity or traumatic event or medical intervention, surgery can give the opportunity for an anaerobe to cause an infection. So this is a good box from your book, kind of illustrating a few examples. Some indications of anaerobes that could be possible in infectious processes. If the infection is close to a mucosal surface, a presence of a foul odor, you might hear an experienced nurse or physician say something like, "Oh, it smells like an anaerobe is causing the infection." These guys smell worse than anything that you have experienced yet. So they smell awful. They produce a lot of gas. There can be a presence of a black color, either on a fluorescence to the organism or the infection itself, or the tissue itself could be black, indicating that the tissue is dead. The presence of sulfur granules should clue you into a certain organism. And then finally some distinct morphological characteristics in the gram stain itself. So we'll talk about a couple unique characteristics that we might see in a gram stain, and you want to be sure to link those characteristics to these certain pathogens. So some frequent ones we're going to talk about are Clostridium species, Bacteroides, Fusobacterium, Prevotellum porphyromonas, Veillonella, and Peptococcus. So some of these, and probably more than not, all you really need to know is the gram stain morphology. Is it a gram-negative rod, a gram-negative cocci, a gram-positive cocci, gram-positive rod? So keep that in mind and keep those in line with the objectives. That way you're not studying more than you absolutely have to. But you can't learn too much about Clostridium. It's probably our #1 genus, and you probably have heard about these without knowing it. So all of the Clostridium species are spore-forming gram-positive bacilli. Okay, they produce spores. Their Clostridium perfringens is a boxcar shaped, and that should say gram-positive bacillus. That's a typo. It produces a double zone of beta hemolysis. So remember that. It's bolded, so it's probably a good clue for the exam. Clostridium tetani is unique. It can swarm on blood agar, and it also has terminal spores, and it's described as almost looking like a tennis racket with these big swollen ends on the terminal ends of the bacillus. So they're located near the ends of the bacillus. Okay, so sometimes where the spores are located can clue you into a certain species of Clostridium. But we're not going to focus a whole lot of that. But I would definitely know that Clostridium tetani and link it with a tennis racket. Tetani, tennis, terminal spores, right? So here they are. We've got Clostridium tetani on the left. Here's a great example right there of the terminal spore. This over here is described as a subterminal spore, so it's not quite on the end, but it's not quite in the center. And that's kind of an off-the-wall species of Clostridium we won't talk about. Some infections, definitely be able to link the disease with the type of Clostridium or the species of Clostridium. So we talked a little bit about Clostridium difficile, and this causes endogenous infections, which means the organism comes from inside your body. A lot of us do have Clostridium difficile in our intestinal tract, but opportunities arise for the organism to cause infection. So maybe heavy antibiotic use, broad spectrum antibiotics can cause the normal flora to die off and Clostridium difficile to over replicate. The production of a toxin could occur, and that's what actually causes the diarrhea or the pseudomembranous colitis. All right. And that's why we use a lot of hand washing and alcohol wipes in the laboratory to prevent the spread of these spores, because we don't want them to spread in the healthcare environment especially. Now, some of these infections can be from exogenous sources, so somewhere from outside the body. So those are like your tetanus, your gas gangrene, botulism, and other food poisoning diseases. So tetanus is caused by Clostridium tetani. That should be pretty easy to remember. And what you really want to remember about Clostridium tetani is that it's associated with muscular rigidity. So the jaw and the neck and the lumbar and the limbs could also be affected. They become very rigid and stiff. All right? Whereas Clostridium botulinum or botulism is flaccid paralysis, so the body becomes very limp. So be able to differentiate those two different types of diseases and those two different organisms. So botulism is caused by an ingestion of the botulin toxin, very, very potent. We can see infections caused by home canning or meats. Infections can produce symptoms within 2 hours to 8 days after ingestion. Infant botulism was seen with contaminated honey. That's why you're not supposed to feed your infant honey anymore. Myonecrosis or gas gangrene is associated with Clostridium perfringens. This is associated. We can see tissue necrosis. That's the preliminary characteristic or symptom that we see. So a lot of gas is produced, and you can see in the picture that the anaerobe, the infection is occurring under the skin, and the anaerobe is producing a lot of gas, and it's actually making the skin swell up into these bubbles, and the tissue is black, right? That's one of our key characteristics for anaerobes. And if you probably could be in that room, it probably smells awful. And that's another clue that an anaerobe is causing the infection. Here is the double zone of hemolysis for Clostridium perfringens. We can see the colony slightly here, and we've got a clear zone just outside the colony here, and then we have another partially clear zone on the outside. So that's your double zone of hemolysis. Definitely know that characteristic. We talked a little bit about Clostridium difficile, toxin A and toxin B, and how that causes pseudomembranous colitis. So we're not going to talk about it a whole lot. To test for Clostridium difficile, we usually don't culture. We would use either PCR or an immunoassay to detect. Actinomyces is a long, thin gram-positive rod. It's branch-like. It can grow in a mass. They kind of appear stuck together. They can be beaded or banded looking. So here it is. Notice, very long. Almost looks like a big chain of cocci, right? That's the beaded gram-positive rod. Here it is again. Here they're grouped up a little bit better. They kind of almost stick together. Very long. Its characteristic colony morphology is described as a white molar tooth. So the colony kind of grows like it's heaping on top of each other. It's normal flora in the respiratory tract, GI tract, and genital tracts, and it usually causes infections in immunocompromised people. So we see these actinomycosis occurs, we see masses in the sinus tracts with discharge, and we see these characteristic sulfur granule colonies. Okay, so make sure you target those sulfur granules when you're setting up your cultures, because the organisms are inside those sulfur granules. Here's a picture of the molar tooth colony. You can see it kind of looks like the top of a tooth, and it's kind of like heaping on top of each other. Here is a person with actinomyces in the sinus tract. Difficult to see, but you can kind of see an indentation there, or you might have a big swelling in the jaw region. Typically, you'll have that big swelling, a big knot in the jaw. Propionibacterium acnes is the number one organism. It's a very common isolate. It's found all over the body, all over the skin. Causes acne. It's a club, pleomorphic, palisading gram-positive rod, right up there with the Colonibacterium, very similar to it. It's described as the anaerobic diphthroids. So typically doesn't cause infections, but it can if it's given the opportunity. So here is some characteristics to note during the microscopic exam. I think this is pretty straightforward. We're going to skip over that. Here's an example of the different types of growth that you would see. This is a pretty good slide. It's a little complicated at first when you first attempt to look at it. So here is some broth from a wound culture, or an aspirate from a wound culture, and the organisms that are in this aspirate are E. coli, Bacteroides, Prevotella, Staph aureus, and Peptostreptococcus anaerobeus. So you have two sets of plates. You have your aerobic set of plates, and then you have your anaerobic set of plates. And you have selective and differential media, and you have selective and differential media over here in the anaerobic plates. So a good experiment or exercise for you is to review what each of these plates are for. What do they do? What kind of organisms do they grow? How do they appear? So here we can see E. coli and Staph aureus will both grow on blood agar incubated aerobically. And here on Brucella blood agar incubated anaerobically, we'll have all 5 organisms will grow on this one plate. Why? Because 3 of these organisms are anaerobes, so they will grow, and these 2 organisms, E. coli and Staph aureus, are facultative anaerobes, so they will also grow if you incubate them anaerobically. So all 5 will grow here, whereas only the 2 facultative anaerobes will grow on your blood agar, incubated aerobically. Chocolate agar, again, on your aerobic plates, you'll only get your facultative anaerobes, right? These are selective plates, so only gram-positive cocci or gram-positive organisms will grow on PEA and CNA, whereas MacConkey will only grow gram-negative rods. Now, if you go over here to your anaerobic blood plates, review which types of anaerobes and facultative anaerobes will grow on each of these plates. So here we have Bacteroides fragilis will grow on BBE. Does it grow a particular color? What grows on KVLB? Here we've got Bacteroides and Prevotella will grow on KVLB. So when we are looking at our cultures, we're taking into account the growth on all of these plates and correlating the growth on all of these plates when we're assimilating our workup. Specimen quality is very important for anaerobes. We talked about this in class. We've got to make sure that we transport it very quickly because we do not want oxygen to destroy the sample, or drying out of the sample can also destroy the quality. How we work up anaerobes is important. There's different ways of working them up in order to keep them away from the oxygen. Be sure to know the unacceptable types of specimens for anaerobic workups. On your direct microscopic exam, this is your gram stain. On your direct gram stain from your sample, it can help to rule in and rule out pathogens. It can determine if there are a polymicrobial infection or a monomicrobial infection. So it'll tell the doctor if there are many different types of organisms present, if you see lots of different types of gram stain morphologies on the gram stain, or if you see one individual morphology. That's very helpful for the physician. And it can also aid in the selection of culture media. If you see a certain type of morphology, you might think of a certain organism and set up a special type of media. There's some unique morphologies when you're performing your gram stain. Number one, we methanol fix instead of heat fix, which is putting a few drops of methanol on the slide and using that to fix it instead of applying heat. We need to recognize unique morphologies like gram-positive spore-forming bacilli. We want to think of Clostridium. Very thin gram-negative bacilli with tapered ends. We want to think of Fusobacteria. Tiny gram-negative cocci that are gram-variable. We want to think of Veillonella. So here's a good chart from your book, just talking about different organisms and media. Some indications of the presence of anaerobes. If you hear some of these clues, it's a good clue that you're dealing with a possible anaerobe. Double zone of hemolysis on blood agar. What organism is that? A brick red fluorescence on KVLB agar. What organism is that? A unique morphology on the gram stain. If we see gram-positive rods with terminal spores that almost look like tennis rackets, what organism could that be? When you're processing colonies that you suspect of anaerobes, the first thing you want to do is describe the colony morphology and correlate the results and the growth on the different types of media. Take into account the gram stain reaction, and then you want to set up an aerotolerance test. You want to make sure that that organism is a true anaerobe, because you don't want to work up the same organism that you worked up in your aerobic culture. You don't want to work up a Staph aureus that's present on both your aerobic culture and your anaerobic culture. So first you want to make sure that it is in fact a true anaerobe or an obligate anaerobe. So what that entails is subbing that colony to two different plates and incubating one in aerobic air and one in anaerobic air. Another thing you might do is use different antibiotic discs, which we're not going to talk about, to help in identifying the organism. Or you could set up some kind of an automated method, which is probably more likely. So here's a schematic when you're determining which path you're going to take or which route, which is the likely pathogen you're working with. Be sure to know the gram stain reactions for the organisms mentioned in this PowerPoint. Be able to interpret an aerotolerance test. I think this is a pretty straightforward slide depicting an aerobe, a facultative anaerobe, and an obligate anaerobe. Here's a good slide showing the difference between 4 different types of anaerobes. A fluorescence is a useful characteristic to evaluate several organisms. Porphyromonas and Prevotella will have a brick red fluorescence when you shine a UV light on them. Fusobacterium and Clostridium difficile is described as a chartreuse fluorescence, and Veonella has this red fluorescence. So here's an example of that. Some other tests that we could use to identify some anaerobes is the catalase test, same catalase test that we've been using. The indole test is same principle, it's using a different reagent, so you have a different color being produced if it's a positive. And then the motility test, which is same principle. Nitrate disk is determining the ability of the organism to reduce nitrate, and the bile disk is determining if the organism is able to grow in 20% bile. And if it is, it's probably this Bacteroides fragilis group. Lecithinase is helpful. We can use egg yolk agar to determine if an organism produces lecithinase. If it does, it will cleave or cut the lecithin found in the egg yolk agar, and this results in this fat that's produced, and it's so like a opaque zone that forms around the colony. So Clostridium perfringens is an example that is lecithinase positive. So link egg yolk agar and lecithinase with Clostridium perfringens. Lipase is another one that we can use to determine if an organism is able to break down triglycerides in a media, and what we see is a multicolored sheen or iridescence to the colony growth on the lipase agar. Here's a slide just kind of depicting some cellular morphologies and some colony morphologies on some of the more common anaerobes and some of the biochemical tests. I would probably stick to just the Gram-Stain reactions and a couple of the biochemical tests for the Clostridiums in particular. Lactobacillus is an organism that's associated with Bacterella vaginalis. Gram-negative rods, that's going to be your Bacteroides species, and your number one pathogen here is going to be Bacteroides fragilis, and we can spot him because he is bile tolerant. He's able to grow in the presence of bile, so we use BBE agar to selectively grow him. He can also grow very large on KVLB agar as well, so he'll be black on BBE and he'll grow on KVLB, gram-negative bacilli. For Prevotella and Porphyromonas, they are not bile tolerant, so they will not grow on bile media. All right, they're gram-negative coccobacilli or bacilli. Prevotella has a brick red fluorescence, so does Porphyromonas. Fusobacterium, key characteristic here is the tapered ends, very, very thin gram-negative rods with those pointy ends. Your anaerobic cocci, we've got a gram-positive cocci, Peptococcus and Peptostreptococcus. So those should be pretty easy to remember. They're pretty similar in names to Streptococcus. And then your oddball is going to be your gram-negative cocci Veonella. All right.